Students of second sem Botany Honours. Today I'll discuss with you all about the primary structure of a dicotyledonous stem, which is included in your syllabus of course 3. So let's proceed. The higher vascular plant has a large variation in the arrangement and structure of primary tissues in stems, roots and leaves. Now when we look into the primary structure of the dicot stem, we have seen that it has three tissue systems. It has the dermal tissue system, which forms the epidermis, the vascular tissue system, which forms the vascular bundles, and the ground tissue system, which includes the cortex, pericycle, medullary rays and teeth. Now the outermost protective layer is called the epidermis. In the epidermis, the cells are compactly arranged without any intercellular spaces. They are rectangular or tubular in shape, and their primary wall is very thin. The outer surface of these cells is covered by a cuticle. In certain cases, chlorenchyma cells are present, which contains chloroplasts and hence helps in carrying out photosynthesis. This is a diagrammatic view of a transverse section of a dicot stem. Now you can see, as I have told you earlier, the outermost layer is the epidermis, and next to it is the hypodermis, and then there is a massive cortex which is made up of parenchyma cells, and this is the central part. Central portion is the pith, and in the next to the cortex you have the vascular bundles arranged in a ring in the round tissue system, and in between the vascular bundles this region is called the medullary rays. So the central portion is the pith, which is parenchymatous, and radiating from the pith there is the parenchymatous zone in between the vascular bundles, which is called the medullary rays. Next to the epidermis is the cortex. That is, the tissue which lies between the epidermis and the stele is the cortex. It is made up of different types of cells. It may be formed of thin-walled parenchyma cells only, as in Salicornia or in Pelargonium, or it may contain other types of cells which lies immediately below the epidermis, and it is called the hypodermis. Now these cells which are present in the hypodermis, they differ in origin from the epidermal cells and structure from the cortical cells. So now we'll discuss about the hypodermis. It may contain peripheral chlorenchyma cells as bands as found in sunflower stem, or it may be present in groups in the ridges as found in cucurbita stem. Whatever it may be, it gives mechanical support to the stem. Now, plastids may be present at the peripheral chlorenchyma cells. Sclerides may be present as in Trachodendron. Oleoresins are present in gingiva. Laticifers are present in Calliotropis. Besides this, it may also contain starch, crystals, etc. Now this is the cellular diagram of a sunflower stem, a dicot, which is a dicot plant. Here you can see the epidermis, cingulate epidermis. Then this is the collenchyma, which is present, collenchymatous region, which is present as a band. But if you look into the cucurbita stem, this is the diagrammatic view of the cucurbita stem, which is also a dicot stem. Next to epidermis, you have the collenchymatous zone, which is present in groups. Here it is present as a band, but here it is present in groups, the collenchymatous cells beneath the ridges. Next to the collenchymatous hypodermis, there is a parenchymatous cortex. Here also you have the parenchymatous cortex. Next to parenchymatous cortex is the starch sheath. Here also you have a starch sheath. Now next to starch sheath, there may be a sclerenchymatous zone. In sunflower, it is present above the vascular bundles as a cap, whereas in cucurbita, the sclerenchymatous zone is present as a band throughout. Next to it, you have the vascular bundles, which I will talk to you about it in my next slide. Next to endodermis, next to the cortex is the endodermis. It forms the inner delimiting layer of the cortex. The entire vascular bundle remains surrounded by the endodermis, but in certain cases, as in nymphia, the individual vascular bundles are encircled by an endodermal layer. In this case, the stele is called polystele. In the endodermal cells, abundant starch grain may be present, giving rise to the starch sheath, which is found in fasciolas. The endodermal cells have Casparian strips. Lignin, suberin and cellulose are deposited on the transverse and radial walls of the endodermal cells as strips to form Casparian strips or band. Now, what is the function of endodermis? Usually, fat, starch, protein granules and tannins remains accumulated in the endodermal cells. The Casparian strips prevent or restrict the movement of water, iron, lead and other minerals in either direction. It also serves as a barrier for pathogen since these cells contain quinones. The cork cambium or phelogen originates from endodermis sometimes, as in Pidaria fetida. Sometimes these endodermal cells behave as meristematic cells, which is termed as proendodermis. They divide to form cortex. Next to endodermis is the massive stele. It consists of vascular bundles and ground tissue system. It is encircled by the endodermis. Now, the pith or medulla, the central part of the stele, is made up of parenchymatous cells, which forms the ground tissue. The vascular bundles actually lies within the ground tissue in between the cortex and the pith. It may be continuous or it may be a split cylinder. The bundles in the dicot, the vascular bundles in the dicot stem are conjoined and collateral, that is, the xylem and phloem, they lie side by side and on the same radii. The phloem is usually present on the external side and the xylem is present on the internal side. The cambium lies in between the xylem and phloem, so it is known as, the vascular bundles are known as open. Sometimes, as in cucurbita stem, the phloem is present on both the inner and outer side of xylem, so it is called bicollateral vascular bundle. There are two patches of cambium as well in cucurbita. The internal phloem is called intraxillary phloem. Now, when these vascular bundles are arranged in a ring within a common endodermis, it is called monostele. But when each vascular bundle is enclosed by an endodermis, it is called polystele, as found in Nymphia. This diagram shows the difference between a collateral open vascular bundle and a bicollateral open vascular bundle. Collateral and bicollateral vascular bundle are found in dicot stem. Collateral open vascular bundle is found in sunflower stem, whereas bicollateral open vascular bundle is found in cucurbita stem. Now, here in collateral open vascular bundle, there is a strip of cambium, only a single strip of cambium, as you can see, and above it, that is towards the periphery, phloem is present, and towards the pith, xylem is present. Whereas in bicollateral open vascular bundle, there are two strips of cambium, and in between the two strips of cambium, there is xylem. Phloem is present above, that is the outer phloem, which is called the outer phloem, towards the periphery, and there is another patch of phloem, that is the intraxillary phloem, which is towards the pith. So there are two patches of phloem. Now, if you look into the xylem vessels, the larger metaxylem vessel lies towards the periphery and the smaller protoxylem vessels lies towards the pith. Here also in cucurbita we find the same sort of arrangement, that is the metaxylem vessels with larger inner space lies towards the periphery and the protoxylem vessels with less inner space lies towards the pith. Now, this sort of arrangement of xylem is called endark arrangement, which is found in this dicot stem. So, you have hopefully understood the internal structure of a dicot stem. If you all have any queries, you all can always ask me through the college portal. Thank you very much.